So thank you very much for the invitation. I am really, really excited to be here, even remotely. And I have to say, my last visit at NYU was at Courant, maybe 3 years ago, something like that. And I had a blast, not only because of NYU, but because I think New York is one of my favorite places on the planet. Yeah, I am very, very biased, I have to say. So today, I want to talk to you about something that you're all very, very familiar with, and that is flow networks, which is more or less the bread and butter of my group. And you're very familiar with the systems because they are everywhere and they span multiple length scales, like from our microcirculation, which is microns, to whole rivers, which are kilometers. And if you look at these two pictures, you will see some similarities immediately. You will see maybe hierarchy, you will see loops. Of course, one is 3D, one is 2D, but again, you might see those similarities and you might think perhaps there are underlying principles. Now, what I would like to convince you in this talk is that there are other similarities that you do not see in these static pictures. And those similarities fundamentally are due to dynamics. And you don't see it because these are static pictures, right? And dynamics, I would like to convince you that dynamics are very important for how these networks develop and how these networks even function. And since I am Greek, I get to show off a little bit my Greek knowledge and my ability to read Greek. And I will read you a quote, "Ta πάντα ρί," from Heraclitus o Ephesios, which basically was conveying the philosophical idea that nothing is stationary, everything is becoming different with time. And he used to say that you cannot step into the same river twice because the river is always changing. Now he was referring to the flow as if the flow was moving, but I wanted to say that specifically with regards to flow networks, this includes the flow that is moving, this includes the properties of the flow, the dynamics of the flow, and this also includes the channel width. And the form of the function of the flow networks I will be talking to you about is very, very tightly linked to dynamics, to the dynamics of the network. Now, as I said, my group's bread and butter is networks, and we do a lot of stuff. And these are some of the projects we've been looking at, not all, but some of the projects that we've been looking at in the past something like 3-4 years, which includes fluid storage in leaves to resilience in networks, many things, topology. But more and more, as time progresses, we're realizing that the time component is very, very important. Now, all of these systems, to a most fundamental and basic degree, from rivers to vascular networks. I do not know if you can see my cursor. Can you see my cursor? Because it seems like
no,
you cannot. Let me see if I can fix this. Because perhaps I'll do this. I'll unplug the big monitor. So, okay, is it okay now? Can you see it?
I see the slides, but I can't see cursor.
The cursor. Okay, and I do not know. I think something is not working well. Anyway, I don't even have Zoom controls, believe it or not. Yeah.
That's all right.
Yeah. So I cannot point, unfortunately, but perhaps I don't need to. So at a very basic level, I can linearize whatever happens in a flow network, and I can describe it as a collection of links, as a collection of nodes, junctions connected by links, and the links are what carries the flow. So there are several parameters several quantities, several fields I need to keep track of, and that is the current through its edge, which I typically use the letter I, the net current that comes in or goes out at a particular node S, the conductance of the particular edge, the pressure. And of course, I will have mass conservation at the node. So if I were to sum the net currents, if I were to calculate the net current at each node, I will get a field S, which will be signifying the net current at a particular node. And I also need a constitutive relation, which tells me how the pressure drop at the two ends of each link and the conductance, how from these two quantities I can get the current. And of course, if I linearize that relationship, I get Kirchhoff. I get that the flow is equal to the conductance times the pressure drop, or if this was an electrical current, instead of the pressure drop, you would say the voltage. So this is essentially the workhorse. Now I will be using that workhorse, it's very, very basic workhorse, to discuss some vignettes of network dynamics. And the first two topics, let's say, that I will be discussing are actually very, very similar. They don't look that similar, but they're very, very similar. And it's about microvasculature and reverse. It's about networks dynamically evolving with time. And the second part of the talk is about networks harnessing dynamics, harnessing activity essentially for their function. And we'll be talking about the necessity for having a pulse. And you're going to tell me, of course, I need to have a pulse because if my heart is not pulsating, I will die. But I will elaborate what I mean with that. And also how to climb pressure gradients with valves. And these are problems related to arteries, veins, and lymphatics. So, okay, there we go. So this is actually a cool movie. It's old, it's from 2003 or so. And it is a cool movie because it shows you something very mundane. This is an image of what would happen to your egg that you have for breakfast if it was fertilized and if it was going to make a chick embryo. So you see in the beginning of the movie, and it's going to loop, you see something like a disorganized capillary plexus, a disorganized network. It's difficult to see because of the resolution, but it's there. As soon as the heart starts pumping, this network gets starts remodeling. You start having some vessels, small vessels disappearing, and other vessels making a big structures, these hierarchical structures. Now, these are some pictures from the paper I took it from so that you can see better what I was talking about. This is the beginning, and I suspect my cursor started appearing now, right? Anyhow.
Yes, it's here. Yes.
Yeah. I think the computer got unstuck. And this down is what happens after the heart has been pumping for a while. Now, the reason for this difference is that the flow, as soon as the heart starts pumping and you have flow, this flow regulates development, regulates the arterial venous differentiation, the chick embryo yolk sac. Now, how exactly that is done, we have some idea. We don't know all the details, but we have some idea of the pathway. And a major player is the wall shear stress. So the vessel wall measures the shear stress. If the vessel is small, if the vessel has a small diameter and a lot of flow through it, you're going to have a lot of shear stress. And the vessel will sense that and will grow, right? Now, from that, I can write down an effective equation. I can actually write down what the equation is for the wall shear stress and translate it to currents, conductances, and all that. And I will get an equation that effectively look like this, where CE is the conductivity of a vessel. So the rate of change of the conductivity is a positive function of the flow. If you use the vessel is going to grow. But if you don't have any flow, that vessel will disappear. And this is an adaptation equation, which is I can essentially use to describe how a vascular network develops. And now, if you see that equation, it only uses local information. Each vessel knows its own conductance and its own current. It doesn't explicitly know what the neighbor is doing. It only knows itself, and it adapts based on local information. Now, this equation, this adaptation equation, was popularized in papers, in a lot of very nice work on Physarum polycephalum, which is a slime mold. It is actually a macroscopic unicellular but multinucleate organism. It's a foraging organism that makes these vessels that you see here in this yellow picture. Now, if I were to add this extra field, which is essentially spontaneous production field, if you were to remove the flow and remove the conductance, then you would have, at that limit, you would have that the rate of increase of the conductivity would be constant, would be that spontaneous production term. Then I get the equations that give me leaf vascular development, although I have to say here, parenthetically, the current is not actually current and the conductance is not actually conductances. It has to do with pin protein flows and auxin canal and auxin concentrations and all that. I'm not going to get into details with that. But basically, what I wanted to say is that this equation is very, very powerful. And actually, we're going to be revisiting it again. So now with this adaptation equation, which again, it's only local information, I will be using it to describe vascular development in actually humans, how you can have a tissue with some capillary network growing. And the way I'm going to be doing this is that I'm going to be having a network, I'm going to be sprinkling conductances, and then I will be calculating the currents. And essentially to do that, it's pretty standard. I will just solve Kirchhoff's equations for this. And I will calculate the currents, then I will apply the adaptation equation, and I will incrementally modify the conductances. That's basically the feedback loop. With the new conductances, I'm going to calculate the new currents. With the new currents, I'm going to incrementally modify the conductances according to this use it or lose it rule, and so on and so forth. And now I'm going to add growth, and I'm going to add growth by saying my input and output, whatever fits the network and whatever takes the current out, the amount of blood in the network will be exponentially increasing with time, with a particular time scale. And that's the growth term.
Yes, Eleni, I have a question on this adaptation model. So Kirchhoff laws, I get where they come from. They come from Poiseuille law, but where does the adaptation model come from?
Yes. Sorry, I'm having technical problems here now. The slides won't even move. There we go. Sorry, I think I have to do this for Zoom. There we go. Let me try again. Sir, play again. Okay. So this adaptation equation is for Physarum. It's phenomenological. For the arteries and veins, I don't have a slide for this, but I can write it down in the whiteboard at the end of the talk. I can write down the shear stress for the, I can write down what people believe is the actual rule, shear stress versus increase in diameter. I can translate what shear stress I expect for particular flow to a term that looks much like the flow term that I have here in this equation. And I can translate the diameter to a conductivity or conductance. And I can get something which is an equation which is a little bit more complicated than this, but it is functionally equivalent to this. So this is where the adaptation rule comes from. For Physarum, it's phenomenological. For the arteries and veins, you can almost derive it. But again, let's consider it phenomenological.
Thank you.
Okay, sorry, I'm having some really difficulties with slides here. Okay, so good. So yes, so the we do that, and we start. In fact, my former student Henrik Ronellenfits did this beautiful work. So we can start, we can actually run this model with growth, because, you know, tissues are growing, and we have a source, and we have this capillary bed that you see here on the left, and we start at 0 time, and then as we see as the time is progressing, we see indeed, we recapitulate what's happening in the egg yolk. You have the system spontaneously differentiating to a hierarchical network. You have the small stuff disappearing, and you have some vessels being selected to be big and some to disappear. Sorry, my computer is completely stuck. And in fact, I missed this slide in the process, which is it's gonna be important for later. So this equation, this phenomenological equation that we saw earlier was not new in a way. I said it was popularized with Physarum, but in fact, physiologists knew it from the 1800s. And in fact, this is the first source, the first person I saw in the literature discussing it, which is Richard Thomas, who said, blood vessels widen with flow. And essentially, this is what this phenomenological equation tells you: blood vessels widen with flow. Of course, there was no Kirchhoff, there was no Kirchhoff quantification in the system, but the idea was there. So, okay, so now I'm going to worry about the following. I'm going to worry about the fact that the dynamics of the boundary conditions of where the blood wants to go is not constant. I talked to you about net currents, the input and the output, but that doesn't have to be constant. There is variations, right? Let's say I need to develop tissue in my hand. I move my hand, I squeeze my hand, you know, the net flow that's coming to my hand will change with time. So I can take this into account, and I can consider an ensemble of scenario of blood input and output, and I can adapt the network based on that scenario, right? So here I have the blue is the input, the red is the output, and it changes with time. And then I can assume that my network measures those currents and adapts based on those currents, right? And depending on the statistics of where the blood wants to go in the tissue, depending on the type of fluctuations, I can get a very different result. So how these fluctuations behave in a way, I can get something which is very loopy, and I can get something which is much more tree-like. So if I have very intense fluctuations, I get something which is loopy. If I have more broad, if the system does collectively, let's say if the fluctuations are not so wild, I get something which is more tree-like. And in fact, I can use this equation, which you see at the top left. And I have, if I non-dimensionalize things, and I remember a few years ago, Sura and I will have this very long discussion about whether we should non-dimensionalize things. But in this case, if we non-dimensionalize the equations, we see we have 3 parameters that we need to worry about. One is how strong the fluctuations are. The other one is the time scale, how fast the network adapts. And the last one is how strong the adaptation is compared to the constant production term that always want to build vessels. That G in this equation always want to build vessels, and this A, let's say, wants to adapt the vessels. So with these 3, we can play with these 3 parameters, and essentially we can create a spectrum of networks that are optimal in doing different things. And we will see that if we look at the functionally optimal networks that are created by these 3 parameters, everything in the end falls onto a line where on one end you have the tree archetype, which is a low-cost network, but it's not robust and it's hierarchical. And it looks very much like our arterial and our venous tree, right? That the network at the large level. And on the other end, we have the chicken wire archetype, which is robust, it's very loopy, and it's also uniform. And these are the capillaries and also everything in between. And the interesting thing here is that a single developmental equation produces a spectrum of complex architectures if you consider dynamics, if let's say biology knows to take advantage of the fact that the flow is dynamical. So this is work that I think the second paper on this work, the last paper was published 2 years since 2 years ago or so. And since then, we've been thinking a little bit on the side about another problem. And that is not only vascular networks remodel and change over time, something that happens in infancy, but also it happens as an adult, like our vascular networks remodel all the time, especially at the small scales. Right? Rivers also change over time. And in the back of our minds, we accept this. And we say, of course, rivers evolve. But it might take eons, it might take thousands of years. And I love this example because it's something that happens in a human lifetime, right? It's the wax delta, which was actually an accidentally man-made delta because the Army Corps of Engineers dredged the Wax Lake outlet in maybe 80 years ago. And they got a delta out of nowhere, which is about here. It's you see in the Google map, it's near Louisiana. And that network is still evolving today. So we talked about Richard Thomas, and we talked that blood vessels widen with flow. And also we have Da Vinci, who said that blood vessels, or rivers, I should say, are like blood vessels. The Earth's blood is the veins of the waters. So we're gonna add these two, and we're gonna use our phenomenological adaptation equation for rivers. And we're gonna see whether we can predict basic things about river, about geomorphology, and in fact about delta geomorphology from this equation. Now I'm going to revisit this equation a little bit because now it is purely phenomenological. It is, yeah, it's purely phenomenological. Now in the context of rivers, the conductance of the river would be perhaps the width of the river. Now you understand that if there is a lot of current in a channel, there's going to be erosion, and that channel will widen. But also if there is no flow, eventually the river will disappear. And this is what this equation means in this context. And there is this parameter gamma, but nobody asked about it yet. So before you ask, I will tell you what it is. This is like an architectural exponent. And it for plants and animals, it comes, one can predict it. It's a scaling exponent that can be predicted from optimization principles. And for rivers, one can look at what the river looks like at steady state and how the conductance, how the width of the river scales with the flow. And from that, one can predict the exponent for the river. So when dCdt equals 0, then I know that the conductance will scale as I to the power 2 gamma. And we have data for rivers. So these two quantities scale for rivers. And from that scaling, we get that 3/5. Now, another observation is much like...
Eleni,
yes.
Can I ask you something?
Yes.
I apologize. People say that my microphone picks some noise, but I will be quick. How do you treat the sign of this current in development equation? If you have a vector through which current can flow in both directions.
That's an excellent question. In this particular case, I don't have to worry about it because the sign, I square it and the sign disappears. So the sign for the systems that we're looking at, and that's not always the case, but for the systems that we're looking at, we are assuming that the directionality of the current in the vessel doesn't matter, only the absolute value of the current. So I can run the
Is
system.
it always the case?
No. No. And in fact, we're going to start looking at, for example, I'm going to jump to the end a little bit. For valve development, it's not the case. One has to be careful because there are symmetry breaking things. But for the level of description that I'm doing here, this is okay, this is enough. And there is another actually aspect, which is really cool. There is another asymmetry so far from what I said. Let's say for the circulation, arteries and veins are identical, so the system should be absolutely symmetric. But the truth is there is another term, and this is a paper I'm not going to talk about today at all. There is a blood oxygenation term. There is a hypoxia term in this equation. And because of oxygen absorption through the bloodstream, there is directionality matters, because if you are upstream, there's going to be more oxygen available than downstream. But now I'm ignoring that, but it matters actually. So yeah, this is an approximation. Okay, so I will continue. So another observation that is actually a little bit counter to our intuition is, or to what we learn at school. I mean, I personally learned at school that tidal deltas have loops, that do not have loops, I should say, that are tree-like. So whenever I learn to draw deltas at school, it looks like a tree, like the left side of the slides. And it's a pity I can't show you my cursor. On the right side, though, we see a real delta, and the real deltas actually have islands. They have loops. So you can take a boat and you can go around fresh water, river water, without arriving at the sea. So where do these loops come from? Previously, in the previous section of the talk, we saw that fluctuation induces loops. So we're going to be inspired by that, and we're going to model rivers the same way. We're going to model rivers like microcirculation and add in fluctuations and see if we can produce the loops. Now you're going to ask me, where do the fluctuations come from? And my answer will be simple. The fluctuations come from tides, because there is, we have the water from the mountain, and we can say this is a constant source. You see it on the right, that's the red, the source of the river. It's a fixed flow. But I also have the ocean, which I call discharge nodes. But what happens is that in practice, the boundary conditions, the pressure boundary conditions of those nodes is not constant. The tide comes in and the river swells. And if the tides are strong, you can even have backflow. And the ones of you that lives on Manhattan on the eastern side, the ones of you that see the East River, it's actually quite interesting. When I arrived at Rockefeller and I would see the East River from my office, and then I thought it was a river, but then I would see the river going this way and then the opposite way. This is because it is, the influence of tides. Now, for this particular project, was driven by an undergraduate, Adam Conklin, fantastic, absolutely stellar, phenomenal undergraduate, who is now a Churchill Scholar at the UK. And we thought to keep it simple. So instead of actually modeling the whole dynamics of how the pressure-driven tidal wave comes in the river, we thought we'll do something very much simpler. We're going to put effective sources and sinks in the network, right? So you see the blue nodes are effective sources and sinks, which we're going to create like, they're going to create the effective pooling of water or the effective decrease, local decrease of the water, right? They're going to effectively create the fluctuations that we're looking for. So we do that, and we start with a domain, which is in this particular case is triangular. It doesn't really matter. And we use the adaptation equation, and we run it in time, and we see that we start for something which is not very well organized in the beginning, and we end up with something hierarchical in the end. And we do that, but we do that by for different scenario, for different amount of tides. And there is one parameter which is called the relative tidal flux. And it is essentially the tidal volume, how much tide comes in every cycle, how strong the tide is, because the tide is not always the same, divided by the input at the source. So there are networks where there is no tides. These are deltas that are not tidal, and that T is 0. And there are deltas that are more unusual, at least for a person that knew nothing about deltas as of a year ago. If you ask a geomorphologist, they're going to tell you they're very usual, where there is no input at the source, there is no river. There is just tides that create a delta. Water is coming in from the sea and back. And you see for very low T, there are some loops, few loops, and you see them noted in purple. As the T increases, when T becomes around 1, maybe 10, you get a maximum of loops. And as you go to tidally dominated deltas, the loops disappear again. And there are many ways one can count loops. In this particular case, we just calculate the average number of loops in simulation. So that's on the y-axis, and on the x-axis is that T number, the relative tidal flux. And you see there is a maximum in the middle between 1 and 10. Okay, this is fine, this is theory, but we want data. And so we went around in the world, and actually I went nowhere. This was the middle of the pandemic. I was on my chair, but John Swank from Los Alamos, and John Shaw from Arkansas, the two geomorphologists in the project, they collected a wonderful data set of networks around the world. These are not all the networks that we had. I just put them on Google Maps, put some of them on Google Maps so that you see them from all over the world. Arctic deltas, deltas from South America, North America, Africa, whatnot. Lots of deltas. And we calculated how loopy they were. So John Swank digitized them, we vectorized them, and we calculate them how loopy they were, and the various metrics of loops. It is, it's all consistent. It's not, how we measure the loopiness will not affect the result. And we measured them versus this T, which is this tidal influence, right? This, how strong the tides are. And we see this is these are the green dots, the deltas are the green dots. And the green dust line is the bind average. We see the effect that we were expecting from the simulation, which is shown in blue. We see that low tides have low loops, intermediate tides have lots of loops, and strong tides have low loops again. And you'll tell me, this is very noisy, and I say yes, because this is the simplest model one could ever make. I disregard everything that has to do with the domain of the delta, with the geography, with what the kind of rock this is. Like, this is hugely simplified delta. And I'm not using an accurate model for the fluctuations either. Nevertheless, I see this trend. And just to give you a feeling, you'll tell me maybe you would have gotten this result by chance. If I were to just randomly permute the deltas, the loopiness of the deltas, I would get this effect low, high, low, only 3% of the times. And the more delta we add, the more that number drops. So this is something which looks like statistically significant result. Now I'm gonna skip this kind of quickly, but the domain, really the size of the domain, the shape of the domain doesn't really matter. And also, if I add noise, because so far the way the tide swells in the my tidal delta is noiseless. It all coherently comes in and goes out, comes in and goes out. If I were to add noise, then the number of loops that I predict will go up. And will go up. It's good, right? It will help my trend. It will help my blue line, which is the theory, go closer to the green line, which is the experiment. Having said that, the actual perfect way to do it would be to actually simulate the tidal wave itself. Anyhow, I want to show you a little bit now some pictures. This is, oops, this is, sorry, I need to move this. Technical problem, sorry. I will remove this here. So this is this here is the Wax Lake Delta, which we saw in the beginning. This is very few tides in the Wax Lake Delta. And this is the prediction for that T. You see only one loop and you see the reality on the right. This is now the Orinoco Delta, which is in South America. And you can not only see the loops on the right, you can also see that the shape of the loops is qualitatively similar to the shape of the loops we predict, and they're kind of elongated from the source to the sea, let's say. And this is my favorite. This is the Ras Issa Delta, which is in Yemen, I think. And what you see, it's difficult to see from the Google image. And I have the vectorized, thresholded image of part of that delta on the top right. And what you see is a little bit like an inverted delta. You see a lot of the, you see like the terminal nodes of the tree being inside the land, and the big branches, the thick branches being at the sea level. And you can also see this on the left on the simulation, these multiple little trees with leaves at the top part of the triangle. So the simulations not only predict the loopiness, but they also predict qualitatively what will these networks look like. So with that, I don't know if there are any questions. Perhaps I should pause here.
Eleni, can I
Yes.
ask one more question, please?
Of course.
Your distributions look qualitatively symmetric when horizontal axis, the tide height or activity of tides, is plotted in logarithmic scale. Do you have
Yes.
any physical feeling of why that's the case?
No, no, that's a very good question, but we didn't look at this so much because we were more interested at the time to match it match it better to the physical deltas, to the real deltas. And in real deltas, it's not so symmetric. It looks like it's not so symmetric. This is an excellent physics question, but we decided not to look at it so much. There's probably a good reason for it, but it's not entirely obvious.
Yeah, that means that somehow twice the tide and half the tide plays the same role, or 10 times the tide and 1/10 of the tide.
Yeah. Yes. This plays the same role in the amounts of loops that we observe. So, but we didn't. We had to choose what to look at, and we decided not to go that way because we wanted to look at. We wanted to add noise, and noise breaks that. If you, I can go back to the plots right here. You see, noise moves the curve. So we're more interested in those effects. But it's very interesting. But we had to, yeah, decide where to go. Okay, so if there are no more questions.
I have
Yes.
well, you still have a ways to go. Yeah. The adaptation equation you used is very similar seemingly for living things and for erosion, right?
Yeah.
Even with similar exponents, a gamma, right? How come? I don't get that. Completely different processes, right? That are giving you that. Is there something, you know, about the form of the equation and the values of gamma where there are limits of gamma where it will, things will blow up or things will,
Yeah.
something will
Oh,
go
yeah.
wrong?
This is an excellent equation and an excellent question for an excellent equation. Why this equation is excellent? Because it is the, in a way, the simplest form you can write down for systems that have all these extra terms and still capture the physics. I love this equation because it is so stupidly simple. Now, actually, the flow term is, in reality, doesn't have that form. Because, of course, you could, if you have, let's say, infinite flow, there has to be some upper cutoff. You can't have an infinite effect on dCdt. So you're kind of assuming you're operating in a range that makes sense, and in a range where that exponent, that I to a certain exponent, captures your physics. But what is very, very important is if I am to look at what type of gradient descent equation, if I were to write down a functional that would give me, and I were to do gradient descent on that functional, and I would get this equation. If I were to, let's say, think of this as an energy landscape where I do gradient descent, that gamma there, the exponent on I is related, and I would think of gradient descent means I have an optimization problem. That is relates to the convexity of the problem. And if gamma, there is a characteristic, critical gamma, above which the problem changes convexity and the problem physically becomes very, very different. And that problem, you stop having economy of scale. What do I mean by economy of scale? In the systems that I have been describing so far, it pays to bundle. If I have a big vessel, perhaps I should annotate my own talk now, but okay, I'll describe it. It is difficult to do this here. Maybe I'll try to share a whiteboard as well. Hopefully you can see it. Let's say I want to reach these three places. One way to do it is like this. But if it pays to bundle things, I can make a bigger vessel that can carry more flow efficiently up to a certain point, and then switch to these three vessels. So I can, if this was, if this has a certain conductance, let's say conductance C naught, then I can have something with less conductance here and bigger conductance here, and that would be preferential. I can distribute my material. So here in this type of system, it pays to bundle, and that means I have a gamma that makes the problem non-convex. And so that is a class of problems that both vascular systems and rivers belong to. Otherwise, you wouldn't have this channelization of rivers. You wouldn't have, the rivers would look very different if the erosion equations that make them happen had a different gamma. So I hope this answered what you were looking for. And let me reshare my slides. Was that what you were
It's
wondering
getting there. I
about?
still don't quite get it. I can see if it's an optimization problem, I can see how it could have been solved by living things by evolution, but I don't understand
It's
how a river system would do it.
Yeah, it is. It happens. This is how the erosion equations work. So I cannot tell you much more about these equations. I can point you to papers, but essentially, if you have two parallel, the simple way to say it is that if you have two parallel streams that carry a certain flow, if there is nothing stopping them, they will merge, and that same flow will be carried by a single channel, because yeah, it's a long explanation. Yeah,
Go on.
it's a very good
I don't want to
question.
hold things up.
So I guess I can go.
Cool.
I only have 10 minutes, so I guess I will just fly through the second part, although it is, in my opinion, even more fun than the first. So now I'm going to switch gears, and I'm going to talk about the work of Son Fancher. And the work of my postdoc, Son Fancher, has to do with the following problem. With the problem that we have a heartbeat. And not only we have heartbeat, but we have an elastic vascular system. And every time our heart is pumping, we have a pulse traveling through the vascular system. And the heart is pushing the blood out, and the arteries that have elastic walls will expand. And when at the next cycle, at the next half of the pressure wave, the arteries will contract again, and so on and so forth. And you can see this in this movie a little bit. Now, we were thinking with Son about optimization in this type of systems with elastic vessels. And I had asked him to run an equation like the one I was showing you before, an adaptation equation, so that we minimize the dissipation in the system. And he came back to my office and he told me, no, this is not working. I don't understand, because if I do this, if I ask this question to the computer and do this optimization, I end up having hyperelastic vessels next to the heart, and the flow, which essentially kills the pulsatility, and the flow in the rest of the system becomes steady. And first we thought that there was a bug with the program, and then we realized there was a bug with ourselves, that we were, like the computer gave the correct answer to a stupid question that we should have seen. It's kind of obvious. Now, we know from basic E&M that you have a pulsatile pump, right? So let's say you have pulsatile flow through a vessel. The dissipation of that pulsatile wavefront is actually higher than the dissipation of a steady flow with the same mean flow. And your body should care about the mean flow, right? So we were asking the computer to minimize the dissipation, and the computer was killing pulsatility. The pump was still pulsatile, and then the system would put a huge elasticity into the next to the heart. Okay, and I want to go to the previous slide. Sorry, there we go. It would make these arteries here next to the heart hyperelastic, and that would kill the pulsatility, right? So we became intrigued by this problem. So we started thinking about it. And to make a long story short, if one writes down the Navier-Stokes for a vessel and does the right approximations, of course, kind of linearizes it, which is something that we have to do as physicists, the equations end up looking like the equations of a transmission line, where essentially the resistor translates to the fact that the flow is viscous, viscosity of the fluid, the inertia is an inductor, and the capacitance are the elasticity of the vessels that can store energy when they extend. And there are various characteristic length scales, a time scales one can look at, and because I don't have time, I'll just give you the basic physics. Right? Now, let's say I have a single vessel. And at the end of that vessel, I have a heart. And the heart in the beginning is not pumping. And then suddenly at time 0, I have this step function of flow happening. What this transmission line was going to do is that it was going to propagate that step function. And the step function is going to become a sigmoidal with increasingly broadened an increasingly broadened sigmoidal. And the speed of this propagation actually is very important for our circulatory system, as we realized, because that speed of propagation lets you know if your heart starts pumping at twice the speed, your capillaries are not going to feel it immediately. How this pulse moves and how this pulse propagates is essentially controlling how fast your body, if you forget that you have a neuronal system, right? How fast your body will respond to a change in the source. We looked at this problem at the single vessel level and shown this wonderful job analytically essentially solving these equations. And there are two limits, the long and the short vessel. But again, I will skip all that because we're out of time. But what he found is that the characteristic time, which we call the response scaling factor, has a very robust form, regardless of the frequency, regardless of many things. In the form, you can see it on the cartoon, on the plot on the right. And what this characteristic scaling function tells you, essentially, what that characteristic scaling function predicts is essentially how much time it takes for if you were to do this experiment here, if you were to send a step function to your body, right? And if you went to, let's say, the middle, if you went to your capillaries, after how much time the flow at the capillaries will have reached 50% of the final flow. So it is, you can think of this intuitively, and I'm skipping all the math, unfortunately, because I'm out of time. You can think of this as an adaptation time scale. What's more interesting is that you can do this for networks. And here I'm showing videos from very simple networks, but we've done it for many networks that are primarily tree-likes, like the one I saw, but you can have many different scalings on how the networks bifurcate. You can have loops, no loops, you can have many, many things. And this form is robust. This response scaling function, where for small networks, it is constant. And then for large networks on the left, it linearly increases. Going back to this, small vessels, short vessels, it flattens out. Long vessels, the longer the vessel, the longer the time will take for the information to reach you, or the more vasculature you have, the longer time it will take for the vasculature to reach you. But even more interestingly, I can plot this response scaling function as a function of the dissipation. And now these plots are quite interesting, because what you see there is, again, very different networks, different how they bifurcate, if they have loops, no loops. But you see very robustly the following trend. The red dots correspond to vessels that are very stretchable, where the elasticity of the vessel is very, very high, where that wind Kessel effect that I showed in the beginning is very high. Networks like essentially what our simulation was predicted in the beginning. Networks that have very high compliance. Now, these very high compliance networks have low dissipation, as our simulation was predicting, but they also have very high response time, which means that my heart would start pumping twice as fast, but my capillaries would feel it after half an hour, right? So you lose the response time, you gain dissipation and you lose the response time. On the other hand, you have stiff networks, which are very responsive, but then the dissipation goes up. And now in preliminary data that I'm not showing, it looks like our own circulation lives at the cusp where that, at the cusp where you go in the plots that you're seeing in front of you right now, the slope changes from being -1/2 in this log plot to being vertical, which means that it looks like our circulation is optimizing the response up to the limit where it starts hurting dissipation. But again, this is speculation. Now, I think I am squarely out of time, despite my technical problems. Okay. Now I am completely frozen. I had another story to tell you, but I don't have time. And it is about the venous and the lymph system, and it's about valves, but unfortunately there is no time. In which case I want to finish by thanking my group, and this is from actually May 2020, from the middle of the pandemic, and everybody looks very miserable. And this is another picture from when we were still live in November 2019. And there have been a lot of changes in the group, and actually we have even more stick figures right now. But anyhow, these are the people that are absolutely fantastic, and these are the people that did all of this work, especially Sean, whom you see here in the middle of the winter with the flip-flops and the sunglasses, and Aaron, who is a stick figure on the right, and also Henrik, who is has left the group a while now, and he is after a stint at MIT, he is now faculty at Williams. So with that, I would like to thank you for your attention. And again, I apologize for my computer who was misbehaving. And I am ready to take questions. Thank you very much for coming, of course, this virtual talk.
Thank you very much, Eleni. That was very exciting. So I think we'll have many questions now, right? Let's continue the nice discussion that Paul started during.
And it was very painful, I have to say, because my computer was very unresponsive, as we saw. I had no cursor. I had a lot of handicaps. I don't know what happened to my system. Maybe it's trying on the background to do an update. I don't know.
So I wanted to ask something along Paul's question. So it's very surprising and somehow like counterintuitive that systems now, be the blood vessels or plant circulatory system or something, but basically being small, so low Reynolds numbers and high effect of surface effects, right? Versus gigantic rivers where the volume effects dominate and you're at high Reynolds numbers. So somehow like I'm like totally bamboozled that at the end of the day it's the geometry and you get actually very similar effects.
Yeah, I have to say though, I am kind of overselling it, because if you look at this, you're gonna tell me, but they look so different. They are, I mean, yes, they have some things that are similar, but there are all these details that are different, to which I will respond, of course, and because there's all of these parameters, all of these higher order terms that I am ignoring, they produce these differences.
Mm-hmm.
But for me, it is the beauty of the subject is that you can find something that essentially captures the big effects, the important effects. And that something is very simple in a way, is that flow will erode, flow will make channels wider, and in the absence of flow, you have no channels. It is that salient aspect. And of course, there are a lot of improvements one that can do to, if you're going to compare with data, you need more terms if you want to compare with data and have a good agreement. And these terms are going to be very different in microcirculation, in micrometers, and rivers, which are kilometers.
Right, right. Oh yeah, it's amazing. Irash.
Yeah, I had a question. You had one slide where you were showing us.
We can't hear you. We lost your audio.
How about now?
Now.
Yes.
All right, sorry about that. So you had one slide where you had this continuum.
It again disappeared, Irash. Your audio goes away every like.
Okay, now it's okay.
It is really close to your mic or yeah.
Yeah, it's probably that.
Okay, so you had
It went away again.
Maybe you can type it.
Yeah. Sorry about it. Maybe Sophie can ask in the meantime her question while Irash types his in.
Sure. I think maybe my question relates to a bit your comments, Alexandra, but what I was wondering was, to what extent is like some form of a Kirchhoff law fine at high Reynolds numbers?
That's an excellent question. It is not. It depends on the effect that you are looking at, very much. So whatever equation you're looking at, I can go back to here, and I can take this, whatever it looks like, and I can linearize it. And if the linear approximation works well enough, then I can be happy. Otherwise, I have to worry. And having said that, we have studied cases where this is nonlinear, and you get very interesting phenomenology. And in fact, there are instances where even in our own circulation, where that F there is not linear, because the vessels, several systems in our body, there are pressure, conductance, feedback. So the vessels, essentially because of that, you have vessels that do not obey Kirchhoff's law. They obey something else. And then also you have valves.
Yep.
And it is in many occasions, linearity is not, even if the flow is laminar and developed and all that, there are many cases where this is not linear as well. But we haven't done, I can't tell you in detail what happens if you have turbulent flows, to be honest. We haven't looked, we look at relatively small Reynolds numbers.
Thank you.
And we have a question from Irash now in
Yes.
chat. So you had a slide where you were showing us a continuum from the blood vessel network of the whole body, tree-like, to the blood vessel network of the capillaries, loopy. And you told us some parameters control the transition in those geometries. But the blood vessel network is one system. Is there a way you can recover this transition from considering some kind of a more general model?
Yeah, that is a good question. So if I understand the question well, you're referring to this slide and you're saying that I might have this transition not only in blood circulation, but in other systems. That's what I understood from this question. That is a good question. The thing is, and we thought about it, I won't lie, what we were interested in at the time is to understand this G here, this constant production term. And we thought a lot about geomorphology, whether that term is relevant for rivers, for example. And it became very contrived. So it wasn't, we had a hammer and we wanted to find nails. What we did find, though, is perhaps this is related to leaf vascular development, which was here, because as I said, this equation with the G that is not showing up here can explain leaf vascular development. It is essentially the plant, if there is any plant biologist or plant biology adjusted person in the group will have heard of this. It is one of the accepted models of how leaves get their loops, get their veins. And what this means in that context is that if you have fluctuations in a certain morphogen, you get leaves with loops. And if you don't have fluctuations in that morphogen, you get more ancient types of leaves that they did not have loops. But in general, we did not think about this spectrum that Iraj, you mentioned, that hard. We just think, it's cool, and then we moved on.
All right, but Iraj, if you have any follow-ups, please. Yes. Thanks. Comes from Iraj. Thank you, Eleni. Sophie, you have. Oh, that's your old hand.
Yeah,
okay.
Sorry, that's my old hand.
Good. Does anyone have a new question that they would like to ask? All right, so if we don't have any other questions, please, let's thank Eleni once again all together.
Thank you very much for having me.
And please stay around if you would like to chat now